The footage exists. Researchers have spent decades watching it. A cuttlefish settles onto a patterned surface, and in under 2 seconds the skin rewrites itself. Color, contrast, three-dimensional texture, all of it simultaneously, in real time. The surface beneath it vanishes. The animal vanishes with it. But the cuttlefish has one photoreceptor. One. The human eye uses 3 types to distinguish millions of color combinations. The cuttlefish has one with peak sensitivity at approximately 492 nanometers, and by every definition in sensory biology, it is completely colorblind. It cannot distinguish hues of equal brightness. It has never seen color in any meaningful sense of the word, and yet the pattern it is projecting onto its own skin matches the color of the surface beneath it closely enough to defeat predators and defeat researchers, not approximately, with an accuracy that should be biologically impossible. That is the question this animal has refused to answer for decades, not how it changes color, how a color-blind animal matches color at all. The cuttlefish belongs to the order Seabed, a lineage that split from the broader cephalopod class approximately 100 million years ago. It is a mollusk without a shell, a cousin to the octopus and the squid, and one of roughly 120 recognized species. They span the Indo-Pacific, the Mediterranean, the Eastern Atlantic, and the West African coast. Almost every coastline on Earth except one, the Americas. Both coasts, the Gulf of Mexico, the Caribbean. The entire Western Hemisphere is a cuttlefish void. The squid fills the ecological niche there. Nobody fully understands why the divide is so absolute. Every species carries the same internal structure that gives the group its name. The cuttlebone, a porous aragonite chamber system, roughly 93% hollow, concealed inside the mantle, not protective armor, a precision buoyancy instrument. By manipulating the gas to liquid ratio in its chambers through osmotic ion control, the cuttlefish achieves near-perfect neutral buoyancy at its chosen depth without muscular effort. It hovers, motionless, stalking prey from centimeters away. The largest species, Sepia apama, the giant Australian cuttlefish, reaches over 50 centimeters in mantle length and 10 kilograms. Comorian fishers have long dried the cuttlebone under the name Hai Piao Xiao as an antacid and hemostatic agent in traditional Chinese medicine, a use backed by its calcium carbonate content. Artisan jewelers still use carved cuttlebone as a casting mold for molten silver and gold, a technique used by silversmiths since antiquity. Pet suppliers sell it globally as a mineral supplement for birds. The cuttlebone has a structural ceiling. The chambers implode under pressure. Research on multiple sepia species found collapse thresholds at around 200 meters, depending on species. There are deep-sea octopuses. There are deep-sea squid. There are no deep-sea cuttlefish. The instrument that makes the cuttlefish the finest shallow-water hover predator in the ocean permanently bars it from depth. And then one species decided to solve the cuttlebone problem by abandoning it entirely. Metasepia pfefferi, the flamboyant cuttlefish, found across the Indo-Pacific from the Philippines to northern Australia, carries a cuttlebone so reduced it cannot achieve sustained neutral buoyancy. Rather than hovering, it walks. It ambles across the seafloor using its outer arm pairs and ventral mantle lobes as walking appendages. While every other cuttlefish in the order defaults to camouflage, the flamboyant cuttlefish broadcasts waves of red, yellow, white, and purple rippling continuously across the mantle, a constant advertisement rather than concealment. This is aposematism, warning coloration, because the flesh of this animal is toxic, not venomous in the bite and inject sense. The muscle tissue itself, research by Mark Norman and colleagues confirmed a toxicity comparable in lethality to the blue-ringed octopus. The chemical class of the toxin remains unidentified. It is the only confirmed toxic cuttlefish alive, an animal that gave up swimming, gave up hiding, and replaced both with a body that kills whatever eats it, back to the skin of the rest of the order. 3 layers operating simultaneously. The uppermost layer is the chromatophores, elastic pigment sacs, each expanded by radial muscles under direct neural command, each controlled by its own dedicated motor neuron projecting from the central brain. Adult Sepia officinalis carries millions of them. They expand from a pinpoint to a disc roughly 1.5 millimeters across in approximately 300 milliseconds. Brown, orange, yellow, red, black. The brain simultaneously issuing individual instructions to millions of individual cells, directly in real time. Beneath the chromatophores are the iridophores, stacked thin reflective plates producing structural color through optical interference, generating iridescent blues, greens, and golds, and also producing polarized light signals used for private communication invisible to most fish predators. Below those are the leucophores, broadband white light scatterers calibrating overall brightness to match the background. 3 independent layers, the full color pattern changing in 100 to 200 milliseconds, faster than a blink. And none of this explains how a colorblind animal decides which colors to display. The leading hypothesis comes from Stubbs and Stubbs, published in the Proceedings of the National Academy of Sciences in 2016. The W-shaped pupil creates multiple simultaneous focal distances. Because different wavelengths focus at different distances through a lens, the cuttlefish may construct a proxy color map from differential blur rather than hue perception. Red focuses differently than blue. The blurring pattern encodes wavelength without requiring separate photoreceptors. This remains scientifically contested. And then there is something weirder still. Research by Methger and colleagues found the skin of Sepia officinalis expresses the same visual pigment gene found in the retina, in fin and ventral skin tissue. The skin itself may be detecting light, each patch sampling the wavelength of illumination falling directly on it, adjusting locally and parallel across the entire body surface without waiting for orders from a brain that cannot see color. Both mechanisms are unproven as behavioral explanations. The camouflage works regardless. The cuttlefish hunts through the same system. When a crab enters range, a slow wave of darkness rolls continuously across the mantle toward the tentacle tips. This is the passing cloud display, a rolling interference pattern that disrupts the prey's visual tracking system. The crab freezes or mistimes its escape response. Then the biphasic strike, a slow, guided extension of the 2 feeding tentacles toward the target, followed by an explosive ballistic snap of the tentacle clubs in milliseconds, too fast for the prey's nervous system to register before contact. The beak cracks the exoskeleton. The radula rasps tissue. The passing cloud is an offensive hunting display. It is not the diamatic display, which is the separate, sudden, high-contrast eye spot pattern deployed against predators to trigger a startle response. 2 entirely different behaviors that popular coverage has conflated for decades. Different contexts, different functions, same skin. When a threat escalates past the diamatic display, the cuttlefish simultaneously releases an ink and mucus mass into the water column, a pseudomorph approximating its own size and shape, and blanches its body to near white. The predator has a dark decoy and a pale animal presenting 2 contradictory targets. The ink is not a smokescreen. It is a body double-built from melanin and mucus. 3 hearts pump blue blood. 2 branchial hearts deliver deoxygenated hemolymph to the gills. 1 systemic heart distributes oxygenated hemolymph to the body. The oxygen carrier is hemocyanin, a copper-based protein dissolved freely in the hemolymph, turning blue when oxygenated. During jet propulsion, high mantle pressure impairs venous return to the systemic heart, causing cardiac output to drop sharply. Jetting is the last resort. The animal built on perfect stillness is physiologically punished every time it is forced to move at speed. The central brain encircles the esophagus and is divided into at least 30 specialized lobes. The optic lobes alone constitute approximately 65.8% of total central nervous system volume. Then researchers at Cambridge University and the Marine Biological Laboratory at Woods Hole administered a modified Stanford marshmallow test. All 6 subjects waited up to 130 seconds for a preferred food reward over an immediately available inferior one. Performance comparable to chimpanzees, crows, and parrots, animals with substantially larger brains and, in most cases, complex social structures. The cuttlefish has no prefrontal cortex. No cortex at all. It passed a test designed to measure prefrontal function. The same team confirmed episodic-like memory. Cuttlefish remember what they ate, where, and how long ago. Memory performance did not decline with age in tested subjects. Cuttlefish also withhold available prey in the morning when experience predicts preferred prey will arrive in the afternoon, demonstrating temporal planning across hours. Because its nervous system resembles no vertebrate architecture, the cuttlefish has become a model organism in Alzheimer's research, allowing scientists to study how neural systems grow, function, and decline in a brain built on completely different principles from ours. That brain begins forming preferences before the animal is born. Embryos observe prey through the egg membrane and imprint behavioral preferences before hatching. The eggs hatch miniature, fully formed adults that already know what they want to eat, educated entirely inside the shell. The mating system is one of the most behaviorally complex in invertebrate biology. Smaller subordinate males, unable to physically contest dominant rivals, run two separate pattern programs on one piece of skin simultaneously. On the side facing the rival, female mimicking coloration, muted, patterned, no courtship stripes. On the side facing the female, full male courtship display running concurrently. Two independent programs on opposite sides of one body at the same time for two separate audiences. This has been filmed. DNA analysis confirmed sneaker males using this strategy succeed approximately 60% of the time, roughly twice the rate of larger males competing honestly. All cuttlefish are semiparous. One reproductive event, then programmed decline. In octopuses, post-reproductive senescence is orchestrated by the optic gland, a small endocrine structure adjacent to the optic lobe that, after reproduction, shifts its signaling profile to suppress feeding and systematically dismantle the organism. Removing the optic gland in brooding female octopuses abolished senescence and extended lifespan by months. The mechanism in cuttlefish is suspected but not yet experimentally confirmed. If the same architecture operates here, the most cognitively sophisticated invertebrate, demonstrably capable of self-control and dual audience communication, is not dying from exhaustion. It is being instructed to die by its own endocrine system. Here is what most documentaries skip entirely. Delay of gratification in vertebrates requires a prefrontal cortex to suppress immediate reward seeking and maintain a future goal state across time. The cuttlefish has none of that architecture. Its brain is a ring of tissue around a food tube. It passed the test anyway. Either executive function requires far less specific neural hardware than neuroscience assumed, and the prefrontal cortex is merely one evolutionary solution to a problem that can be solved in other ways. Or the cuttlefish evolved a completely convergent cognitive system producing behaviorally identical results from anatomically unrecognizable hardware. Both answers demand revision of foundational assumptions about what intelligence requires. Neither has been resolved. The animal posing one of the most destabilizing open questions in cognitive neuroscience is primarily known to the public as the ingredient in squid ink pasta. The cuttlefish feeds industries across 3 continents. In Vietnam, Japan, the Mediterranean, and coastal China. It appears in forms ranging from grilled street preparations to high-end restaurant menus. The ink, known commercially as sepia, gave its name to a Renaissance era, drawing pigment used since the ancient Greeks for writing and coloring cloth. Global cuttlefish landings reached approximately 348,000 tons annually, according to FAO 2018 data. Most major fisheries in the English Channel, the Mediterranean, and the Northeast Atlantic carry no formal stock assessment and no scientifically determined catch limit. The giant Australian cuttlefish is near threatened on the IUCN Red List. Commercial fishing removed approximately 250,000 individuals from the Point Lowly spawning aggregation in Spencer Gulf in 3 months in 1997. By 2013, the aggregation had collapsed to roughly 13,000 individuals. Following a complete commercial fishing closure, it rebounded to approximately 247,000 by the late 2010s. Recovery was possible because the pressure was removed in time. The IUCN Cephalopod Specialist Group monitors global cephalopod population trends. The Australian Marine Conservation Society works specifically on Sepia apama. The Blue Planet Society advocates for English Channel fishery management reform. Ocean acidification adds a mechanism none of these frameworks yet account for. Juvenile cuttlebones developing in more acidic water form denser, heavier structures that disrupt buoyancy control. The precision instrument, the entire ecology of this animal depends on, may be compromised in the next generation before the animals are born. The octopus carries approximately 2/3 of its neurons in its arms, giving them significant semi-autonomous function. The cuttlefish has a proportionally larger central brain and more centralized neural control, a different architecture producing different specializations. The bobtail squid, Euprymna scolopes, farms bioluminescent bacteria in a specialized light organ to eliminate its own shadow from below, an entirely different cephalopod solution to concealment built on biological partnership rather than skin engineering. The nautilus, still carrying its external shell, is the ancestral body plan this lineage started from 500 million years ago. Set one next to a cuttlefish. The distance between those two animals is the full story of what cephalopod evolution decided to do with time. An 18-month lifespan, a ring-shaped brain, complete color blindness, camouflage that defeats science, self-control that defeats neuroscience, and a death its own endocrine system may be the instrument of. And if you want to dive deeper into the world's most bizarre creatures, hit subscribe. We're exploring the biological impossibilities that science is still trying to explain. And if you thought the cuttlefish was strange, wait until you see what we're covering next.